Great. So welcome and good afternoon also from my side. Great honor to be moderating this session of the Rising Stars together with Tamas here. Thanks to the organizers for this opportunity. Just allow a minute or so just to introduce myself. My name is Michael Hübner. I'm the country lead here in Switzerland for J&J Innovation. J&J Innovation is a global team looking for early stage innovation wherever it is globally. We primarily stand on three pillars. One is early stage partnerships and collaborations with startup companies. Early stage for us is everything up to clinical proof of concept. The second pillar is that we run our own incubators, 13 of those globally. They're called J-Labs. These are no strings attached incubators. And the third pillar, that's our venture capital fund, JJDC. It's a strategic venture capital fund. And with this comprehensive toolkit, we support startup companies and create partnerships with people like you.
And again, also welcome to this very exciting session. My name is Tamas Schweighoer, and actually I'm co-founding partner with a relatively new Basel-based venture capital partnership called 4051 Ventures. We are dedicated for company formation and helping and growing early stage, exciting science-based new companies. So without further ado, then I think we should start. I need to remind the dear speakers that time is limited and you will get some feedback. So foreseen is 3 minutes for your elevator pitch and 2 minutes for questions and discussion. And at the same time, I would like to ask our audience to think about questions, because this is also very helpful as a feedback to the speakers, right? So with that, please.
With that, thank you. With that, we'll kick it off. We have 16 presenters. We'll start with the first 8, which are primarily focused on oncology. So I'm asking to the stage the first presenter, Darren Cunningham from Mysthera from Basel, Switzerland, please.
Okay, always great to be first. Good afternoon. As mentioned, my name is Darren Cunningham, co-founder at Mysthera. And I should mention up front that Tamas at 4051 is on our board and is an investor in the company. So what do we do? So we are hoping to write the next chapter in autoimmune disease therapeutics. We plan to do this by pursuing first-in-class approach to selectively regulate multi-lineage drivers in autoimmune disease, including B cells and T cells. Now, the uniqueness here is further underscored by the fact that we are the only company pursuing PIM kinase inhibition in autoimmune disease. So this gives us a shot at targeting complex autoimmune diseases where unmet need remains, the likes of lupus nephritis, rheumatoid arthritis. And we've already assembled a team with considerable expertise in PIM kinase and immunology. And for example, I have 12 years experience working in the PIM kinase space. Only established this year, so a brand new company. We have achieved a number of milestones. We've raised 3.5 million in seed capital from 4051 Ventures here in Basel. We've in-licensed our preclinical portfolio of PIM kinase inhibitors, and we're now very much focused on raising a further seed extension of 3 million to advance that program. So very briefly on our target here, PIM kinase, we view PIM as a gateway kinase in autoimmune disease. Now, what's interesting for us about PIM kinase is that it's an induced kinase, and it's induced by many of the key players in autoimmune disease like JAK-STAT. So this in effect places PIM at the scene of the crime. So when we deliver our drug, in effect, it's preferentially targeting the disease lesion, thereby sparing healthy cells. And this is really an unmet need in the space. Now, once activated, PIM drives autoimmunity through regulation, modulation of T and B cells. And furthermore, it promotes the interplay and the crosstalk between T and B cells, which is a key cornerstone for the building of these tertiary lymphoid structures, which are associated with more complex disease. So if you take that all together, we feel our therapeutic concept will offer a greater efficacy opportunity in complex autoimmune diseases and better tolerated. This image here, just briefly at a high level, shows the mechanism of action and how it's differentiated, the multi-lineage aspect of our approach versus other existing approaches. And regarding our portfolio, we already have identified a lead asset, and we are looking now to, with the seed investment, to confirm that as a clinical candidate such that the company can commence IND studies towards the end of next year. And I'll leave my contact details here in terms of my email. So if any of you are interested to learn more about the company and our seed extension, please don't hesitate to contact me. Thank you very much.
Great. Thank you for staying in time. We have time for 1 or 2 quick questions,
Excellent,
if
yes.
there are any.
So just a very nice pitch, quick pitch. Just a question on if you go to the clinic, what would be like your phase 1 study, and which indication would be in your wish list?
Yeah, so that's a good question. It's always a challenge when you've got a mechanism like this, that kind of application across a number of different settings. So already we've done some work on this, and we're doing this in terms of understanding where the mechanism of our drug and the biology are a match. So for example, in lupus nephritis, we already know there's a platform of evidence that PIM kinase is relevant. Furthermore, these tertiary lymphoid structures that I referred to earlier are a key component of driving the complexity of that disease. So given PIM's role in those 2 areas, we think a PIM kinase inhibitor could be well suited. So a phase 1 A/B, for example, could be an important clinical step for us as we kind of refine our clinical strategy in the next short while.
Great. Thank you very much. We have to move on in the interest of time. Thank you.
Thank you. Thanks very much.
The next presenter is David Pioski from Adoram from Connecy in Switzerland.
Good afternoon, everyone. It's a real pleasure to be here. My name's David. I'm CEO and co-founder of Adoram Therapeutics. Our aim is to make better small molecule therapies that are both safer and more effective than conventional small molecule therapies. So I can use our lead asset as an example of why allosteric molecules are better. You can see here with this example of A2AR antagonists, those that have been used in the clinic shown there, they have to bind the same site as the natural ligand, adenosine here. And that means when there's too much adenosine, like in a solid tumor, these drugs get displaced and they stop working. So that's one of the causes of the main limitations. They're not potent in a solid tumor, and there's some other problems you can see listed there with this first generation of drugs. So we went and used our proprietary allosteric screening cascade to define allosteric modulators. And you can see in this animation, we identified a pocket that no one else has targeted before. And that means the receptor changes conformation so that adenosine can no longer signal. So we've effectively blocked this immune checkpoint, and the immune system is restored. It can kill the cancer. But you can think of other advantages here. So you don't have to go for a maximum tolerated dose with an allosteric molecule. You can just go for a target coverage approach. And that means you have lower doses, and that's safer for the patients. You've got a better therapeutic window. And some more details about the other assets in the company. I've just told you about the A2AR negative allosteric modulator, the NAM. We have a good patent for that. And secondly, we have allosteric modulators for inflammatory conditions and the screening platform itself to discover new assets. This is a summary comparison with these other drugs that are in the clinic for our lead asset. You can see we're aiming for a best-in-class based on potency and safety. We have shown monotherapy in mouse models, which most of the other competitors have not shown. And what we're asking for now, we're finalizing our clinical candidate drug selection. So we need less than 2 million for that to be able to also scale up. And then next year to do the IND enabling studies. You can see in that top line. We know that from discussions with Pharma, they would potentially partner as early as even before the clinical trial. Most Pharma over here, though, and with our other assets, that would only require less than 1 million to continue. And this is just showing the timeline and what kinds of funding sources we would be going for. So to introduce you to our team in the last few seconds, here you can see we have clinicians. We're also onboarding new advisors, and we have collaborators locally and abroad. Here, just to thank our funding, we've had 2 million francs from non-dilutive sources. And if you'd like to stay in touch, please connect on LinkedIn. I'd be happy to take questions. Thank you.
Thank you for staying in time. Do we have any questions here? See one.
Thank you, David. How exactly are you planning to select the dose?
So this in like a phase 1, we can do the standard 3x3 type of experiment, and going up with allosteric, we're going to also work with a collaborator at the university who can do calculations to enable us to choose the very initial dose, and then after that expand the dose that looks the best for the 1B. I can't, in terms of the details, I'm not sure how they do the calculations, but we have a collaborator who will work on that.
Do we have another question? If not, perhaps I can ask it. It's of course a very competitive field. Did you reach out to VCs and pharma companies yet, and what feedback did you get?
From VCs, we're in due diligence with a few of them. So I'm in pretty deep scientific DD, and then hoping to close that round by the end of the year. We also have some business angels that would like to help us with some more immediate funding. With Pharma, we've reached out to about 20, about 9 of them would like to follow. We have a couple of CDAs, and some of them, as I mentioned, would like to partner quite early, the majority more in the human data phase.
Hello, everybody. So I'm Sam Ducan. I am the CEO of TheraOnco. And at TheraOnco, we have developed a property platform that enhances efficacy of the wide range of payload and reduce the side effect. So as everybody knows, even today, it's very complicated to target specifically all the tumor cell and reduce the side effect. And probably one way to be able to solve this issue will be to increase the number of artificial epitopes on the surface of the cell in order to be able to better target the tumor cell at the end of the day. The way to do it might be using click chemistry and some metabolites that are clickable. This metabolite could enter the metabolic pathway of the tumoral cell and will, at the end, decorate the surface of the tumoral cell. Doing so, we incorporate a huge number of epitopes on the surface of the cell, and we will be able to click by click chemistry any kind of payload. This approach has been used since 2010 by a huge number of academic laboratories, and you have more than 500 publications all around the world using this approach, where they use a sugar, a non-natural sugar that is metabolized by the tumoral cell in the first step, and after they add a drug, and the drug is click on the surface of the tumoral cell and kills the tumor cell. Unfortunately, this approach is even if it's very nice, the problem is that the sugar is very toxic, and you have no company that use this approach today. So it's where we are playing. So our idea was to change the sugar to a sugar that is naturally incorporated in the pentose pathway, because everybody knows since Warburg effect that the pentose pathway is upregulated in all the tumor cells. And by consequence, we expect that the fact that we have this sugar that enters the pentose pathway will be able to be incorporated mainly by the tumoral cell. In fact, it's exactly the case, and we have very nice results in vitro and in the animal, where we are able to tag specifically the tumoral cell and kill after with payload specifically the tumoral cell. So in summary, there are 2 steps. The first step is the capability of labeling the tumoral cell in the human, and after you add the payload and any kind of payload to kill the tumor, based on the fact that we increase the number of epitopes on the surface of the tumoral cell. So we are waiting to find partner, because we are able to improve all the payload, in terms of target efficiency, and reduce the toxicity or increase IP lifetime. And we have full package from proof of concept to license. And we are also waiting for a new investor. We have just started a run that have been, we have just finished a run this year, and we will start a new run next year of investment. Thank you for your attention.
Thank you. Do I see any questions here? Maybe I can start with one for ADCs. Obviously, the therapeutic index is key. Do you have any experiments, any data yet, any predictions? What kind of a therapeutic window you can reach with this technology?
So what we have done is we reduced by more than 3 times the concentration of doxorubicin needed to kill some tumor in the mouse. But we have not still, optimized the process. We have good proof, but not optimized. And in vitro, we are more than 10 times higher in terms of efficiency. And
Thank you.
as you know, you have signed a worldwide agreement with Chasky in the US, and they perform almost the same, but we do it best, to make it simple.
Thank you very much for the opportunity. My name is Itziar Kanamastas, and I'm the CEO of AtG Therapeutics, a company focused on overcoming tumor adaptation. Why is tumor adaptation important? As we know, tumors are dynamic. They change to evade the natural defense mechanisms and ultimately to grow aggressively and spread, leading to poor prognosis of many cancer patients. We have a very clear value proposition. We're developing a first-in-class inhibitor against a tumor-selective enzyme, TA1, that makes cancer cells vulnerable. And we also have a discovery platform against tumor adaptation vulnerabilities. We're looking to raise a C+ round of 7 million that would allow to select our development candidate as well as then build the pipeline. Why we believe we're going to be able to make it happen is because we have a great team committed to make it a reality. We have a world-renowned scientific founder in tumor escape mechanisms with Uriol Casanovas. We also have a great set of operational strategic advisors and a superb advisory board with the likes of Professor Hanahan, which is the cancer biologist in the world, who is also an angel investor in the company, as well as Professor Schlesinger and other great names with Jeff Hager and Dr. Bolak. TA1 is a cytoplasmic enzyme with multiple pro-cancer functions that gives us the opportunity to design a first-in-class intercellular inhibitor. TA1 meets all key selection criteria that we think are critical to develop an inhibitor of high therapeutic index that will address changing tumor dynamics, because we have a biomarker that we can simply test with an ELISA test in plasma samples of patients. We have seen that cancer cells become dependent on TA1 upon anti-angiogenic treatment, a market that is hugely large and a backbone of chemotherapy, as well as immuno-oncology agents and indications such as colorectal cancer, kidney cancer, and liver cancer. But also, many cancer cells, many different tumor types, overexpress the target to drive cancer growth. We have demonstrated in vivo proof of concept that by inhibiting the tumor growth in a resistance setting of high TA1 expression, we are able to arrest tumor growth, both in a gene silencing approach, as you can see here on the left-hand side, as well as then with a tool compound that is a small molecule inhibitor of intracellular inhibition and modest potency. We've also seen that TA1 inhibitor shows first-line efficacy as a single agent in a setting of higher TA1 expression, and also presents with an additive effect in induced dependency upon combination, what we call induced synthetic lethality. TA1, as mentioned, is measurable in plasma, and this will allow for a differentiated survival patient population, which will be key to speed up and de-risk clinical development, because we're going to have already a very selected patient population that will progress fast and where we're going to be able to see a relevant efficacy signal in our phase 1b dose extension trial. We have all key experiments and partnerships in place to drive the selection of our TA1 development candidate by the end of 2024. And with the 7 million C+ funding, we hope to be able then to reach the development candidate selected, also have the target validated, and ultimately have a successful Series A fundraising that will bring us to generate this clinical efficacy signal in the clinic. So thanks so much for your interest in AtG. We really hope to bring this opportunity to many cancer patients and to continue to build a strong pipeline with our tactic platform. Thank you very much.
Great, thank you. Do we have questions? Yes, I see one.
Thank you for the talk.
Thank you.
I was wondering about that. I saw that you're waiting in the clinical trial for a positive monotherapy signal. Are you not scared that you might miss a better positive signal by combining with some other drugs, say EGFR inhibitors?
Yes, thank you for the question. Our approach is not only to do monotherapy. We're looking to first focus on generating a fast monotherapy signal to drive the value inflection point, but parallelly, we would start also a combination strategy, first with anti-angiogenics. For example, if you look at colorectal cancer, you would do a combination with Avastin, which is used in first line, second line, third line, and any cancer line in colorectal cancer. At the same time, you very rightly say that there's a huge opportunity beyond the anti-angiogenic market. The target is overexpressed in many different cancer types, and also we will explore the opportunity of combining with different modalities such as anti-EGFR, etc.
So I'm very pleased to be here today to present a super powerful technology. I've been in the biotech business for several decades now, and as a VC or a panelist, I never such a promising technology. This is the reason why, instead of enjoying life and take care of my vineyard, I decided to create Adoram Therapeutics, which is my third company as CEO. So how does this technology work? This technology is the fruit of more than 20 years of academic work. It is called NutriReg, and it consists in three building blocks. The first one is a proprietary and patented artificial promoter. The second one is a therapeutic gene whose expression is to be switched on or switched off. And the third one is a proprietary and patented food supplement which is able to activate an ubiquitous signaling pathway. This activation results into the release of a transcription factor which itself activates the promoter. As can be seen on the right, when the system is off, nothing is happening. But 1 hour after the mice are fed with a food supplement, the system is switched on and the gene is expressed. And when the food supplement is removed from the cage of the mice, the system is switched off and the expression of the gene is stopped. Because there is no toxicity, we are just talking about amino acids, because the induction is rapid, as you have seen, because there is no leakage and because it is fully reversible, NutriReg is powerful, simple, rapid, and safe. That makes NutriReg the unique platform that enables treatment requiring to switch on, then to switch off the expression of gene. So, where is the need? Gene therapy story started in the 90s, and at that time, companies realized the huge potential offered by this technology for producing therapeutic genes within the body. However, it never developed because it required a robust control system for controlling gene expression in order to avoid the toxicity of therapeutic peptides produced at full capacity 20 hours a day. This is what we bring today. And it will change the face of peptide and shRNA delivery on the face of gene therapy as a whole, because contrary to what exists for animal use, NutriReg does not need any pharmacological inducer. NutriReg is fully human. NutriReg works for any gene expression product, and NutriReg is applicable to any delivery system, whether it be viral or non-viral. Having received a lot of grants during the past few years, we have been able to answer to most of the basic questions addressed by any preclinical development. SAX Forum is therefore for us the first event of a roadshow aimed at raising 4 million in Series A. Those of you who attended my yesterday's speech have noticed that we have convincing preclinical data supporting our strategy and even phase 1 data in healthy volunteers. We strongly believe that this unique, powerful technology will change the face of gene therapy, and this is the reason why we have funded this company. Thank you.
Thank you. Do you see any questions? Maybe I can start with one. The activation mechanism, I think, is a mixture of amino acids. Do you see any risk of patients having a different uptake or metabolism of amino acids that might influence the on and off switch of your system?
Not at all. In order to produce your protein, you need 20 amino acids. However, 3 of those, valine, leucine, isoleucine, are transported from the intestinal lumen into the bloodstream by the same transporter. So if you intake a reduced quantity of leucine on an overload quantity of isoleucine on valine, you saturate your transporter, and the quantity of leucine in your bloodstream goes down very quickly and activates the signaling pathway. So as long as, so if when you eat back a normal meal, let's say a piece of meat, your system is off. So there is no risk of parasite activation because you are not adding something, you are just removing something, which in terms of regulatory issue is much more favorable.
Thank you, Michael. Happy to be here. I'm the CEO and co-founder of CaSRevolution. We are targeting a new mechanism of action to treat Alzheimer's disease. I will show you how it works. We discovered that both on astrocytes and neurons, there is a receptor, which is called calcium sensing receptor, which is able to bind to beta oligomers. This binding activates a neurotoxic cascade both in neurons and astrocytes. That's very important, that also in astrocytes. You have further beta produced by neurons, and in astrocytes you have also further beta plus tau, VGF, and nitric oxide. These newly created beta oligomers are then linking again to the receptors of the original cells, but also the neighboring cells, continuing the vicious cycle. And then the neurons die. What we discovered is that there is the possibility to use a negative allosteric modulator, which is called calcilytic as a chemical class, that is able to interrupt this binding of beta oligomers and the calcium sensing receptor, thereby interrupting the entire neurocascade. So we're talking about interrupting the three hallmarks of the Alzheimer's disease, saving neurons. So basically, our mechanism of action is a novel biology, which is very interesting because we heard big pharma are looking for novel biology, particularly in Alzheimer's disease. This is involved in all the three major hallmarks of the disease. This you see neurofibrillary tangles, amyloid accumulation, neuroinflammation. It's very important to notice that the interaction between beta and the calcium sensing receptor is moving the amyloid precursor protein into a beta secretase, and so that's the reason why it starts the amyloid accumulation. We have plenty of independent data, so we didn't produce the data ourselves. It's a collection of data provided by the literature, both in terms of pharmacological inhibition, both in vitro and in vivo, and also genetic manipulation. So knocking out the receptor, you have you save from neurodegeneration the mice in this case. We just raised 1.6 million from biotech executives in Italy. We are based in Milano, I didn't tell you. Family offices and a seed vehicle from a Pharma foundation. We are now in the process of performing our drug discovery. We just started the medicinal chemistry. We already have the assay, primary assay prepared. In October, we will have the first batch of compounds to be screened, and we hope to arrive at a lead at the end of the spring next year. And then we will begin the optimization phase, which will require a substantial amount of money, also because in parallel, we are beginning to work on the translational models, both tau and beta models. We are now starting to work with Columbia University on brain tissues, GWAS, to expand the clinical evidence that we have so far. And of course, one of the major challenges will be to choose the right patient population in Alzheimer's disease, particularly because with Big Pharma, we're discussing about how to find the right clinical proof of concept for this novel model of action. The team is quite senior. We have a board of directors with people that created and exited companies like Luca Benatti, Andrea Pellacani, who has been an executive in the field for many, many years in the US, and then very senior drug developers that have been working on CNS in the last few years. Thank you.
Thank you very much. Questions in the audience? Perhaps I can ask one. Can you tell us a bit about your IP protection and where you currently are in that regard?
Yes, sure. So we started our computer-assisted drug discovery approach with a knowledge-based approach and discovering the crystallography of the receptor. And so we started with a series of compounds that have been patented as a negative allosteric modulators, but not with claims in Alzheimer's disease, to start our strategy the medicinal chemistry part. Now, the goal, of course, is to have a potent compound which is brain penetrant. The class of compounds historically have been developed for other diseases, particularly osteoporosis, because the calcium sensing receptor is involved in calcium homeostasis. And we know from the phase 2 data of some partners that there is no adverse effects or let's say, other receptors involved with these calcilytics, just a small hypercalcemia. But the compounds currently on the databases have not been developed for brain distribution. So we have to work particularly on this part. So we expect to have the patent ready during the springtime.
Great. Thank you very much.
Hello, thank you very much for the opportunity of being here and to present you Allogenica's activity. I'm a scientific co-founder and CEO of the company, and we are developing the next generation allogenic CAR T-cell therapies because we strongly believe that there is an urgent need to bring these life-changing therapies to as much patient as we can. So our unique approach is based on the physiological development of T lymphocytes, T cells, which normally occur within the human thymus. And what we developed as an expertise is we are able to recapitulate early stages of T cell development in vitro. So we generate immature T cell precursors from cord blood-derived stem cells, and we engineer them to express the CAR. And the main advantage of using this kind of approach is these cells can be injected to the patient, finish their maturation in the patient-dependent manner, and be recognized as patient-own immune system cells. So we developed this expertise as a bioproduction platform for allogenic immunotherapy based on the use of CAR pre-T cells. So it's important for me to place our technology in the really crowded landscape of allogenic therapies. So first of all, we all need to avoid graft versus host disease, which naturally occurs when you inject cells from one person to someone else. And for this, Allogenica's approach relies on physiological thymic education of immature cells were injected to the patients. However, most of the other allogenic techniques require one genetic modification. To avoid rejection by the host of our cells, we are doing banking, meaning that we partially match donor and patient for the application. And most of the allogenic CAR Ts requires more than 3 different genetic modification. We also believe that our cells are more persistent by nature. They are able of long-term persistence. However, most of the allogenic CAR T cells only persist for 6 months. And we also believe in reinforced CAR T efficacy, no expected side effects. We are able to divide our cost of manufacturing by 10, and we have a reinforced security for the patient since we're introducing only one genetic modification, the expression of the CAR. So we have the whole platform, the whole bioprocess internally in-house, and this have some advantages for bioproduction, but also therapeutic advantages, as I mentioned earlier. We established some proof of concept, in vitro proof of concept studies, where we show that our CARs generated internally were able of killing of cancer target cells, but also we show in preclinical animal models that mice injected with CAR pre-T cells show better anti-cancer activity as compared to control mice. Since we have a platform, we're also focusing on the development of 3 different assets. So for our lead candidate is CD19 CAR. Since we are testing the new modality of the treatment, which is allogenic treatment, we go with validated target, which is CD19, for the treatment of pediatric and young adult lymphoma. And today we are looking for the seed round of 3.5 million to essentially finance tech transfer of our bioproduction process towards a CDMO and preclinical IND-enabling studies. But we also use the whole toolbox which we generated to develop first-in-class CARs, allogenic CARs in leukemia and solid cancer. Up to date, we raised 2 million euros, and today the plan is to raise 3.5 in seed and then goes for Series A of 15 millions to finance our first in man clinical trial. Thank you for your attention.
Great, thank you very much. Just perhaps a question from me. Do you see yourself as more of a platform company, or are you focusing on the assets you're developing? How would you interact with a pharma company?
Actually, we consider both modalities of interaction. So first of all, we are today developing assets based on our technology, but we are completely open-minded to work with other partners, biotech or pharma, if they have a target which they're interested in to bring it on allogenic scale.
Just a short question also from me. I think it's a very intriguing scientific idea to bring immature T cells into a new host, but does maturation continue there? And do you see expression of endogenous T cell receptor repertoire, which can then cause graft-versus host problems?
Yes, thank you very much for the question. So basically, we inject IV in animal, in mice, our cells, and they express thymic homing receptors naturally. So they home to the thymus directly, and then they finish and they acquire T cell repertoire but based on patient MHC. So that's why they are becoming like recognized as patient-owned immune system cell.
Hello. Let us do for aggressive cancer what we've already done for COVID. IPSirius is beating aggressive cancers using a new form of immunotherapy, basically using iPS cells as a delivery system for cancer targets to better prime the patient's immune system against their own cancer. This is the team. It's basically me and three founders, the professors in the University of Paris. So there's clinical oncologists, stem cell experts, bioinformatics experts specializing in cancer stem cell characterization. We've talked a lot at this conference about treatment resistance and the need to develop new therapeutics that get around this problem. And some of us might have even seen this slide or this picture in other people's hands. Basically the problem of stopping cancer relapse and treatment resistance. And ultimately this comes down to targeting two elements. One is the cancer stem cell, and also the protective niche in which they reside, so the tumor microenvironment. Our therapeutic product targets both of these things, and this is the rationale for our therapeutic product. iPS cells and cancer stem cells share a common signature in terms of transcriptionally and their epigenome. So unlike adult stem cells, for example, or somatic cells in the patient, you can use iPS cells to prime the immune system against its cancer, or against their cancer, against the patient's cancer. So this is the therapeutic product. One of the reasons I joined this company is it's the simplest iPS cell therapeutic product that I've ever seen. You basically expand the number of cells, irradiate them, add an adjuvant, and that's your therapeutic product. So, I think it's been referenced in the earlier panel that manufacturing complexity in the cell and gene therapy space is always a problem. And this really caught my attention. Ultimately, it's off the shelf, easy to manufacture, and it's an adjuvant therapy that you can put on to the current standard of care. So this is what the preclinical data looks like. In essence, when we inject the iPS cell-based vaccine, you get remission of the tumors, you get a cessation of metastasis, and you also get an extension of the life of the animals. We have an extensive patent portfolio, 29 patents over 14 territories. And so far, we've raised 2.2 million, mainly through the European Innovation Council and BPI France. So we're coming here today as a Series A to ask for 40 million, and that will take us through to the very end of our first in-human trial, with patients with advanced non-small cell lung cancer. And as it was alluded to in the panel as well, it takes a village, and we have lined up key expertises as part of that village, namely the Cell and Gene Therapy Catapult, who's helping with our manufacturing and regulatory pathways, and also the NHS, the Scottish Blood Transfusion Service. So ultimately, our customer is helping us manufacture this therapeutic product. Thank you for your time, and look forward to talking to you.
Thank you. Do we have any questions in the audience?
Yes, we do.
Hi, thank you very much for the presentation. So I've never seen irradiated cells before used like this. Did you have any regulatory feedback from experts on that side?
Yeah, I mean, it's, there's at the Cell and Gene Therapy Catapult, the head of manufacturing there has a wealth of experience generating dendritic, cell vaccines. But this is, and the radiation feeds into that kind of product as well. But we believe that with the iPS cell therapies, it'll be very distinct and much more successful.
But could you imagine then using maybe systems that would precise, there are systems that exist that make more precise cuts in the DNA of those cells. Could you envision using those kind of technologies or not?
You're talking about our pipeline then. So that, yeah, I mean, as long as we get the required amount, there's an endless stream of possibilities we can consider.
All right, thank you very much.
So I'm Helga, and this is my team. Together we fight leukemia, which is my heart's project for the last 20 years. AML is a deadly blood cancer affected 100,000 of patients worldwide per year. Although most of the patients respond to the initiated therapy, 80% of the patients relapse in the following 2 years and die in 5 years. That is why we need very urgently strategies that stabilize the remissions. And that is where we come in, since we provide an effective and easy to apply maintenance DC-based immunotherapy for these patients. So the most advanced immunotherapies right now are based on ex vivo prepared cells. Afterwards, they are manipulated, which is very time-consuming and very complex. And afterwards, these cells are retransfused to patients. And even very often, patients die before their cell preparations are ready. And we do, in contrast, a very different approach, since we only infuse a drug combination to the patients in vivo and induce anti-leukemic cells, immune cells, specifically targeting those cells in vivo. And then in vivo, we get an activated immune system leading to a blast kill on the one hand and to the installation of memory cells that avoid that relapses in the future. So where do we stand right now? So we have done a lot of preclinical research in the last 20 years. At the moment, we have a scientific advice and successful scientific advice of the bee farm. And now we want to start with a phase 1 and phase 2 trial. So, and let's talk money now. So we've based our financial model on the sales figures of venetoclax, which is another drug for the treatment of leukemia. And doing an extrapolation of those data and the ramp up, we come out with 100 million sales revenues if we are successful. So what we would like to get right now and what we want to raise is the money to conduct a phase 1 or 2 trial. And that is what you see here. And for the up to now, our funding is based on us funders and also on some private backers. And now we are looking for 3 million for the phase 1 and another 20 million for the phase 2 conduction of these studies, and then we have an exit option. And if we would get another 41 million, we could start to bring Modiblast to optimize the drug formulation. So this is our project in a nutshell, and I would like to invite you to join us in this venture. Thank you very much.
So, do we have questions in the audience? Because otherwise, my key question is here. I think it's a very intriguing approach that you are converting leukemia cells into antigen-presenting dendritic cells. But in many cases, one or more elements in the antigen presentation pathway is already missing. So can you prove that antigen presentation is still very active? And what are the evidence for that?
Yeah, so what we regularly do is that we check the co-stimulatory markers on the surface. That is the one thing we do. And the other things are not so interesting for us, since the most interesting result is whether we can get the immune system to become leukemia specific and to address the blasts and to kill the blasts at the end. And that is what we can check in different functional assays, and what we regularly do, and what we could prove, and what we are going to publish or have already published.
Thank you very much.
You're
If
welcome.
there are no further questions, then we move on. And our next speaker is Merle Fuchs from PRAMOMOLECULAR from Berlin.
So I'm Merle Fuchs, CEO and co-founder of PRAMOMOLECULAR. 1 in 2 in this room will develop cancer during lifetime, and 1 in 4 of these cancers will be caused by mutated KRAS proteins. KRAS proteins are the most important oncogenic drivers in humans, and most of the mutations are still considered undruggable, because a small molecule drug you can see in blue needs a binding pocket marked in red, and most oncogenic KRAS mutations lack this binding pocket. They are undruggable by small molecule drugs. But you can prevent the KRAS protein, the mutated KRAS protein, from being formed by gene silencing, for example, with siRNA. Why are there no siRNA drugs against pancreatic cancer or lung cancer? Because siRNA drugs normally only work in the liver. PRAMOMOLECULAR has a delivery molecule. We can deliver siRNAs in vivo into the cells of the lung, of the heart, of the pancreas, and also of the colon. Our delivery molecule is very simple. We just couple defined lipids, we define chemical bond covalently to the siRNA. This can be done fully automized, GMP compliant on a standard oligonucleotide synthesizer. And we don't need any particle formulation. Here you see downregulation of the mRNA, of the KRAS mRNA in a PDX model of human colon carcinoma. And you can see that we have a strong efficiency in downregulation, no mutated mRNA, no disease-causing protein, no cancer. And here you see that we can prevent cancer, the growth of cancer. In black, you see the in vivo growth of cancer of human lung cancer in a PDX model, and in red you see if we treat them, we can reduce cancer growth. Currently, we do co-development with another developer of therapeutic oligonucleotides against solid tumor, and we develop our own drug candidates against for 2 systemic applications and 1 local application. We have applied 2 patents for our platform technology, and we will apply additional patents for each drug candidate. In the upper row, you see the founders. All of them are scientists. In the lower row, you see pharma experts who strengthened our team, and I'm a molecular biologist and learned interdisciplinary work in the lab of Nobel Prize winner Manfred Eigen, and I'm co-founder of additional 7 high-tech startups. We are mainly a virtual company, but we work with experts among the best in their field. So we need partners and we need money.
Thank you very much for the clear presentation. Questions from the audience, please. Because otherwise, I just really would be interested to know how you actually selected those specific lipids. And you mentioned you are not targeting liver anymore, but what would be the preferential targeting versus liver?
We did repositioning. We didn't select these molecules by ourselves. But I mentioned I'm co-founder of an additional high-tech startup. They do GMP compliant synthesis, so I was aware about the problem of delivery. And we heard about a professor who spent his whole academic life in developing delivery molecules for delivering mononucleotides. And our idea was to test them for oligonucleotides, and it works with antisense oligonucleotides, it works with siRNAs, and probably it works also with other molecules. We didn't select the specificity. We looked for the organ preference, and then we selected the applications.
Okay, good afternoon, everybody. So the last major improvements in terms of cancer treatment have been brought by the ICI. Unfortunately, only 20% of the patient can benefit from the response to this ICI. So we aim to create a solution that can increase this number and also create de novo immune response, since the responder usually respond because they have a preposition response in their body. So in the field of the cancer vaccine family, we belong to the DNA-based cancer vaccine. However, we have the platform patented also for RNA combination. So what we do is a combinatory approach to develop this vaccine, and we are based on a synthetic DNA. So that's have the big advantage of having no bacterial contaminants, but it's a very short process of production, and it's very low cost of goods. So that's allows us to provide vaccines within a very restricted period of time, 8 weeks, which is very competitive and at a cost that's going to be very acceptable for the healthcare providers. So what we do is we do the personalized DNA based on the selection of the neoepitopes by artificial intelligence and high-speed sequencing. That's the personalized part. The two other parts are amplifiers of the immune response, and that's multiplied by 40 the strengths of the immune response. This is just one product, and it's accepted by the MEA as a TMP product. So we have several proof of concept in different preclinical models that the one have been used by all the competition. So we have very good results in a very aggressive melanoma model, B16, in terms of survival. We have the green curve here. In terms of tumor growth, we have also results in colorectal carcinoma and in 41 breast triple-negative breast cancer. I started the presentation saying that the aim was to increase the level of response to the ICI. So what you see in the green curve as compared to the red curve, which is the anti-PD-1 response at around 20%, we can bring that level to 60%. And we also treated that with combination anti-CTLA-4 and anti-PD-1. And here again, we have good results. We were happy to see that we also have results in monotherapy with the compounds. So the turnaround time is 8 weeks, as mentioned, needle to needle to the patient. And the secure, the thing was always about the manufacturing and the CMC. So we particularly developed the CMC and the securing of the supply chain by locking all our partners. And we're ready to do that even for phase 1 with this timing in GMP ballots already to go to phase 1. That's now we're going to submit the CTA by the end of the year. And we also working on the scale up of so we can treat personalized 100 patients a day. So we need the 5 million to get into phase 1 and then 20 million to extend cohort in phase 2A. So that's the team. That's a team of experts in their domains, and we have a strong advisory board as well. So in a nutshell, so we have a disruptive technology with the use of a synthetic DNA, and we are going to treat, we're going to go to phase 1 in colorectal and pancreatic cancer. And we also can develop the platform within a new solution or other indications. Thank you for your attention. I would be happy to answer your questions.
Thank you very much. Okay.
Perhaps I can ask you a question. I think you went over the slide pretty quickly, where you showed a slowing of the tumor progression. But do you also see a regression of established tumors with that technology?
We saw that with the 41 breast cancer model. Yes, we could see that. We have more data, but in 3 minutes it's difficult to show them all. But we have tested all the preclinical models that have been used by the competition. So we have all the data to support that. Yeah.
Great, thank you.
How do you actually choose your neoepitopes? Because I think this is the real critical ingredient.
Yeah. We have contracted with the company that does that by artificial intelligence, saying that we're not targeting the most frequent neoepitopes, but we're targeting the more immunogenic epitopes. So that explains why we have been disappointing by the previous neoepitope approach. It's not because of the approach itself, it was because of the toolbox. Now we have a lot of data sets, we have a lot of algorithms that are very performant, and that's how we choose the neoepitopes to build the DNA.
Well, thank you, Tamas, for the kind introduction, and thank you everyone for still being here. I was just upstairs coming down, and it's pretty empty upstairs. So thank you. Good. InCephalo. So we're currently working for local therapies, so local, behind the blood-brain barrier, directly into the brain cancer. And these are next generation local therapies where it's all about having 2 key characteristics. Once you apply them locally, they should stay long enough for the drug to make a difference. And once the drug leaves the brain tumor, for example, it should be rapidly degraded, all with the idea of significantly improving the therapeutic index for highly toxic drugs. Quickly about InCephalo, that's myself. I'm a trained biochemist with an economics degree. InCephalo is the 3rd biotech company that I have the chance to co-found and lead. We then have Johannes, who's a translational expert in brain cancer disease, and we have Lauren, who's our clinical expert in brain cancer disease. She actually ran more than 25 clinical trials during her period as vice chair in the neuro-oncology department at Memorial Sloan Kettering Cancer Center in New York. Thanks to her, obviously, we also have access to some of the world-renowned neurosurgeons and neuro-oncologists in the field, and particularly in the US and also here in Europe. I mean, just mention a few here, such as Michael Lim, the chair of neurosurgery from Stanford University, or Nino Giocca from Harvard Medical School, who has a specific expertise in one of the assets that I'll show you later on. So overall, just quickly briefing over that slide, we have all necessary expertise basically to bring a drug into clinical trials and through clinical trials. I mean, given the fact Lauren also has a substantial commercial expertise, but that's maybe a little bit of too much future thinking. So brain cancer disease, as you're probably all aware, it's an extremely challenging disease indication. Nevertheless, the unmet medical need, the burden is huge. I mean, in the US alone, only 100,000 patients each year get diagnosed with brain cancer, and for most, unfortunately, it remains a death sentence. And why is that so? All of you probably know that our brain is a protected space. The blood sometimes gets infected, but the blood-brain barrier makes sure that none of these viruses or bacteria actually enter the brain. The unfortunate truth is that's also true for chemicals and drugs. So drugs don't penetrate the brain sufficiently. So what actually happens if you infuse a drug intravenously, most of it stays in the bloodstream. A marginal amount actually enters the brain and gets close to the brain disease, or in particular, the brain cancer. And this is particularly true for antibodies, where if you're lucky using F-star Denali's or Roche shuttle technology, you maybe get a few percentage, 2-3% actually into the brain. So what actually happens is, how do you get the drug into the brain? Well, let's think of that problem differently. Neurosurgeons are actually applying drugs behind the blood-brain barrier as we speak, directly into the brain. The unfortunate truth is none of these drugs are designed for local application. And we in InCephalo, we create next generation local therapies that actually address this particular problem. The drug should stay in the brain, so the drug has enough time to make a difference at the brain cancer, and it should not leak out into the body to have any systemic severe side effects. We have some data, but then again, we only have 3 minutes, so I don't want to focus too much on the data. But our primary asset has received orphan drug designation, and it's a proprietary so-called compartment lock IL-12. IL-12 has a history in big pharma of being dropped. Why? Because IL-12 leaks very rapidly into the systemic compartment and creates severe side effects. As you see at the bottom left-hand side, for example, when you inject IL-12 either in humans or into mice, you have the whole body gets inflamed, you have interferon gamma spikes, so you have severe side effects. But our compartment lock, as depicted at the top left-hand side, even though there is a brain tumor in the mice, even though the blood-brain barrier has been compromised, the drug stays in the brain. We don't see any neurological toxicity based on our non-GLP tox study, and we don't see any interferon gamma spikes in the body. And that's mainly where IL-12 side effects have arisen. So better compliance is important. I mean, IL-12 has been, for example, worked in clinical trials by Nino, one of our advisors from Harvard Medical School. So better compliance, better tolerability. But we also believe we have significantly better overall survival. Now just depicting basically the key attributes of this slide. We're very happy to see a single agent efficacy, but more importantly, it works in combination with radiotherapy, it works in combination with chemotherapy such as temozolomide, and it works in combination with immune checkpoint blockers. So really, we believe we have found a significant attribute to current standard of care. Just quickly where InCephalo is, our current value inflection points of the current round where we're still looking for 2.5 million Swiss francs. We're de-risking our primary asset with regards to production. We just started CMC production, cell line development, toxicology. We're just having a GLP tox study in rodents. We are having our first regulatory meeting in December this year. And in addition to that, we have a so-called ex vivo clinical trial study. So whenever a patient here in Switzerland, we're working with 4 hospitals here. Whenever they have their brain tumor being resected, we co-cultivate the brain tumor in so-called UCAPs and then look at of the microtumor environment. We look at cytokines and we look at single cell RNA. Plus, we have an additional asset in development. Thank you and sorry for being over time.
Great, thank you for the kind introduction. So we're actually a spin-off of the Psychiatric University Hospital in Zurich, and we are a clinical stage drug development company focusing on what we call regenerative therapeutics for mental health. I guess it's not news that we live in the age of mental health pandemic, with significant morbidity and mortality. Pharmacotherapies so far mostly address symptoms and are not disease modifying. So what we understand with regenerative therapeutics are compounds that have a rapid acting reduction of symptoms and a long-term disease modifying potential. The compound class that promises this is one that we describe as neuroplastogens. So compounds that promote neuroplasticity and actually help the brain rewire. And so where this market is coming from and going, here's a quick overview. So I would say the ice-breaking product is Janssen's Spravato, an S-ketamine nasal spray. Had a difficult takeoff, but now is set on for commercial breakthrough as a blockbuster. And on the heels, we have what is known as street drugs, psychedelics, coming behind. We have MDMA for PTSD. Last week, a second phase 2 published in Nature Medicine with response rates close to 90%, remission rates over 50%. Same for psilocybin for depression, treatment-resistant depression, and even shorter acting compounds like 5-MeO-DMT on the heels. Right, so huge promise. I don't think we need to discuss the efficacy potential here. The challenge is how to square those from a commercial perspective in the healthcare system and how to manage what is perceived as side effects, so the hallucinatory effects. This is something that we have focused on. So we've been in stealth for the past 6 years working on this, coming from an institution with the longest tradition in research in psychoplastogens. And actually, we have solved most of these issues. So just to give you a quick contrast of where the industry is standing today. So we have the generation 1, They go for high dose. It's kind of this magic pill promise. You give them patient once and suddenly everything is fine. It's actually not how it works. Other companies are proposing, okay, let's engineer completely the subjective experience away, focus only on the plastogenic aspects. But anybody who knows psychiatric drugs knows that they are always psychoactive, right? Those that work are actually psychoactive. So we'll see where it depends. We think, as Swiss always do, the compromise is somewhere in the middle. So how do you sharpen the psychological effects so that you harness the therapeutic properties and reduce the adverse events or non-therapeutic properties? And this we do through innovative formulations and precisely controlling the pharmacokinetic profiles, which then yields in a more precise control of subjective psychological effects. And so the approach we take is a fast follower, looking what happens in the market, adapting the duration and route of administration of these drugs to what the market is ready to uptake. With this, we have originated a portfolio of 3 compounds, currently all phase 2 ready. We have phase 1 readouts in healthy volunteers, very promising efficacy surrogate markers, mostly psychological surrogate markers. We have the first compound, which is following an interventional approach of Spravato. The second one, which is focused also on the short-acting 5-MeO-DMT. And the third one is non-psychedelic or non-psychoactive in this sense. It's a classic repurposing 505B2 with a novel mechanism of action for insomnia. And it really promotes healthy sleep. So what we see is a consolidation of sleep architecture, including REM sleep, where natural neuroplasticity actually happens. This is the team. Founders are all clinical researchers, KOLs in the field of psychoplastogen research, but we have staffed up with senior pharmaceutical and biotech expertise. We've raised close to or over $12 million now, over $6 million in grant financing. We're now going out to test the waters for a bigger Series B round and actually have a fallback position for a Series A2 with the lead already committed 5 million plus. So if this sounds interesting, happy to talk to you. Thank you for your time and attention.
Any questions from the audience? Your first combination reminds me of an old Indian concoction called ayahuasca. Are they kind of precedents for that, or are you imitating that in a more pharmacological way?
So if you want a bit more of a nice description of what we're doing, there's just now on Swiss television on a documentary called NZZ Format. That was a documentary about our research group and research we're doing. It is indeed inspired by ayahuasca. We combine a reversible MA inhibitor called Harmine. By the way, it was marketed by Merck for Parkinson's disease with really exciting results, taken off the market because they couldn't standardize the pharmacokinetic profiles. This is what we have solved for around Harmine. That's where most of the IP revolves around, novel solid state forms, novel formulations. And this compound in combination with DMT yields the specific ayahuasca effect, which has been studied in peer-reviewed journals. There is great promise for things like addiction. That's what we're focusing on. Smoking cessation as a showcase. Most people think now of opioids when you talk addiction, but actually still the biggest killer and healthcare issue is nicotine, cigarettes, right?
Okay, thank you very much, Davor.
Thank you.
Ladies and gentlemen, it's a pleasure talking with you today. My name is Urban Skog. I'm CEO of LaRive Bio, a newly founded company in Zug, Switzerland. Our mission is to improve treatment outcome for MS patients. This is Elizabeth, 38 years of age. She's an MS patient. As you may be well aware, there exists no cure for MS today. Treatment is alleviating symptoms and may impact on disease progression. The problem is the side effects affecting the quality of life of these patients, and we may have a solution to that. CLX-106 is a new molecular entity which we have taken on from a research group, and 106 aims to demonstrate equal efficacy and superior tolerability than the current oral first-line treatment for MS patients. In discussions with FDA, we have decided to go with an abbreviated, accelerated, cost-efficient development path to market. This is the way forward. You can see we are at this point seed raise Q4 2023. That is for the next step. That is the proof of clinical concept phase 1 trial that we intend to initiate. We wish to raise $10 million for that. We have identified the inflection points, you can see them above, and also plan for potential cash in points on the journey. How do we operate and who are we? LaRive, as I mentioned, is a newly founded company in Zug. We are setting up a core team with 2 key functions, ClinOps and licensing. We work together with top-tier external partners. And on the left hand, you can see we establish contact with innovative research groups. Some of them are small. Many of them have neither experience nor competence, let alone financial means to take their innovation forward. That's where we come into play. We enter post preclin, and we can take the candidates through phase 1, 2, and 3 and exit before market entry. We select the candidates according to patient benefit, commercial attractiveness, and efficiency, cost and time efficiency in the development. This is Jan Beckman, my colleague over there, and myself. We are the founders of the company and the initial leadership team, which is on its way. To conclude, we are focusing on de-risked business propositions due to application of accelerated development process. We are taking this molecule into a very large market segment. It is a molecule which is promising and has a robust IP. And this is not only for patients' benefit, but also for shareholder value. I thank you for your attention.
Try not to blink, because every time you do, you lose hundreds of cells from the surface of your cornea. I'm just kidding. That's of course completely normal. The reason you're not going blind is because there are cells located here on the edge of your cornea called limbal stem cells. These cells constantly produce new corneal epithelial cells to replace the ones you blink away. But these cells can be destroyed, for example by fireworks or chemical injuries, and this causes a condition called limbal stem cell deficiency. In this disease, the corneal epithelium can no longer heal itself, causing blood vessels and surrounding tissue to cover the cornea. And it's a severe and painful form of blindness, which cannot be cured with normal cornea transplants. Limbal stem cell deficiency is a rare disease with a real unmet need. It mostly affects young and working age people. In fact, the average age of the treated patients is only 34 years. I'm 34. Can you imagine the impact of losing sight at this age? The impact for patients is devastating, but there's also a big impact to the society. Cell and Gene Therapy Catapult estimated that the maximum reimbursement potential for limbal stem cell deficiency is almost €250,000 in Europe and over $500,000 in the United States. And that's for one eye only. There are about 36,000 eyes to treat in Europe and the US, bringing the total market size to 14 billion. These are our competitors. They're doing an amazing job, but they can only help patients who still have one healthy eye left. Patients who are totally blind have no treatments. And that's over half of all the patients. We don't need a donor eye to take cells from. We use induced pluripotent stem cells, which we then differentiate into limbal stem cells. Okay, what's so special about our technology? It's simple. That's it. It's simple. It's simple enough that we can manufacture it at low cost and at scale. Thus, we are solving the biggest problem in cell therapies today: commercial viability. And of course, we have patents and we have the scientific knowledge to back it up. Our team is composed of top scientists in the field, and we are backed up by our board who complement us. They are experts in the pharmaceutical industry, regulatory affairs, and ophthalmology. Now we're raising 7 million. With the funds, we'll expand our own labs and grow our team further. And also we'll get the first product ready for clinical trials. As we expand, we can also go into other indications in the cornea field. I'll leave you with this. We are StemSight. We are the only ones who can actually cure blindness. Thank you.
Thank you, Laura. Any questions from the audience? So I must admit I had some exposure to limbal cell transplantation. And one surprising thing was that these cells are very immunogenic. Now you are using probably patient-derived stem cells?
Actually not. We are going after allogenic iPSCs. So we have a bank of clinical grade iPSCs. But you are correct. They are immunogenic. And that's why we are using state-of-the-art hypoimmunogenically edited cells, where the HLAs have been knocked out.
Very much, very important detail. So, and ladies and gentlemen, any questions? Otherwise, please talk to Laura afterwards.
Thank you.
And with this, I think we are coming to the last presentation of the conference.
Thank you very much. I'll be very quick. I'll skip many slides. So we're FimmCyte and we're developing the first disease-modifying treatment for endometriosis. If you're not familiar with the disease, endometriosis is a painful chronic disease that affects about 10% of women globally. And the disease is characterized by cells that are normally lining the uterus, leaving the uterus and invading adjacent tissue, very similar to what you expect in cancer, but fortunately it's not terminal. But the unfortunate part, it's as painful as many cancers, as many women reported before. And what this means is that the physical life quality is significantly affected in these women. It's a leading cause of infertility, and it's also a leading cause of hysterectomy globally. The significance is that in a country as small as Switzerland, you're looking at 2 billion in direct and indirect costs. In the US, it's exceed 100 billion. And the problem is that 30% of women do not respond to the current standard of care, which is majorly hormones and painkillers. And as you can imagine, this is insufficient given the very large number of women that are affected by this disease. And the other or the final alternative for these women is to completely remove their uterus, as I mentioned. And what we've done is that we created FMC2. It's a novel antibody that is non-hormonal. It's targeting a novel protein that hasn't been described before in the disease, and it labels the cells, the endometriotic cells, for elimination. It has very good selectivity in several studies, as you can see here. In the treated group, we have on the left side, the pointer is not very good, but we looked into ectopic cells. So these are the lesions, the disease. We started from 5 lesions, went all the way to 1 in the treated group, and the untreated group, around 3 lesions are left out of the 5. And here on the right, you can see if you implant the normal tissue that is lining the endometrium, we don't see a difference between the treated and untreated, which is what you want. You don't want to kill any of the healthy cells that are present. You're killing the bad cells and you're leaving the good cells. We started by the support of Inniswiss a couple of years ago, and we spent last year and this year working on vivo model and optimizing our lead. We are supported by many of the early stage investors that left Bioinnovation Institute, Base Launch, and we're also supported by Ferring Pharmaceuticals. Our objective is to raise a seed round next year, hoping to go into humans in 26. Anybody wants to talk to me about this, happy to make the time. And with that, I'd like to close. Thank you very much.
Thank you very much. Questions to that important topic? So I was actually really wondering that endometriosis is usually responsive for hormonal cycles and also anti-contraceptive treatment. So how do you envision to combine these antibody contraceptives, for example?
So the target market we're after is usually women who are not responding to these first and second line treatment of contraceptives, but also anti-GnRH and GnRH agonists. So we're kind of going into last resort. So do you have a surgery or do you go for our treatment? And we're hoping that this becomes then the lead rather than resorting to surgery.
Thank you very much.
Thank you.
There's a question in the audience.
It's very exciting data and also very important the work that you do. What I'm finding very interesting is you eliminate those cells. Do you in the end have a curing antibody or is it a repetitive treatment that you have to apply?
Our hope is that it's curative, but I do not use this word until we are in the clinic and we see some, because the disease model simply cannot tell you any of this. So we need to wait.
Great. So with that, I think on behalf of Tamas and myself, thank you very much to the presenters and thank you to the organizer.